My name is Marina Rautenbach. I'm from Stellenbosch University, South Africa, and I want to introduce you to sticky antimicrobial peptides to create cellulose matrices and polymer surfaces with sterilizing activity. There is a monster called antibiotic resistance. They are currently one of our biggest enemies. It is in our environment, it is in our houses, and sometimes in our body. And we need to take note of this. And if you look at the media, they've already taken note of it. Because drug-resistant bacteria kills thousands of people, if not millions of people, and we are under threat. And it's predicted that we will lose more than 10 million people per year due to drug resistance in 2050. I think it's going to be more than that because we will not be able to cure cancer or do any surgeries without antibiotics. And this is man-made. We use more than 130,000 tons of antibiotics in livestock, and this use is predicted to go up to above 200,000 tons. We use more than 280,000 tons of biocides that can induce drug resistance, and is even more so since the COVID pandemic. And humans use more than 50,000 tons. So obviously the microorganisms are going to fight back. And this is a man-made enemy, namely antibiotic resistance. The resistance spread is the problem, and we can curb it by using less antibiotics. But to change the way that we've been using antibiotics up to now is going to be quite difficult. Is there another way we can stop resistant pathogen transfer and disease transmission? Is we can stop it at the source. And that is where you have direct contact with the organism, droplet spread of it, airborne transmission, as well as vector-borne transmission. So if we can stop it there, we can also limit pathogen transfer, resistant pathogen transfer, and disease transmission. I want to introduce you to sterilizing technology from nature, namely antimicrobial peptides from soil bacteria that we can use for creating functional antimicrobial materials. Some of the natural peptides that we've been working with are the tyrosidines and analogs. And this structure here, for those people that are interested in chemical structures, are the structure of tyrosidin A, a phenylalanine rich peptide. And here we can see that these peptides in general have 4 aromatic amino acids, and this is the ones in green. And the ones that are important that I'll talk about later on would be number 6 and number 7 residue that can either be phenylalanine or tryptophan or both. I chose these peptides because they have a long history of use as topical antibiotic, and they were actually the first antibiotics used in Second World War for topical applications and wound sepsis. And up to now, there's very little documented resistance, which is a good start. And the reason why there's very little documented resistance because they have multiple targets in a rapid mode of action, and that helps against resistance. They have a broad spectrum of activity, and they have high stability. They can handle high temperatures. They're pH stable and UV stable. But what is good for commercial applications is that we can have it produced by microorganisms in culture, and this pertains to a very economical production. In order to combat pathogens on surfaces and create self-sterilizing materials, it is good to have a broad spectrum of their activity. And these peptides indeed have a broad spectrum of activity. They're specifically very active against gram-positive bacteria and filamentous fungi, as well as Candida albicans, which is a pathogenic yeast infecting humans. They are less active against gram-negative bacteria. And just a note, the lower the number here, the more active it is, because the lower concentration you need to kill the microorganisms. But what is interesting in terms of the gram-negative bacteria, for example, the tryptophan-rich peptide I spoke about earlier, are quite active against gram-negative organisms. And we can also add maybe metals and another peptide to improve the activity against the gram-negatives. They also have activity against cancer cells, but I do not recommend that we use these peptides against cancer cells, simply because they also have other toxicities when using them systemically. They are very well tolerated as topical applications, throat lozenges and topical creams. So they are quite safe to use on a regular basis, but I would not recommend injecting these peptides into your body. So how did these peptides avert resistance during the last 80 years? Well, they have rapid membranolytic activity and multiple targets. So on the left-hand side, if you look at these traces, they're basically showing the opening of channels. So when the trace goes up, an ion channel in a membrane is opened and ions are released into the environment. And then in this way, there's a functionality that is lost in a membrane, because the membrane is supposed to keep ions on the inside that's supposed to be on the inside, and ions on the outside that's supposed to be on the outside. The moment these membranes actually lose some of their functionality, this can cause the organism to die as a consequence of lysis, or just losing functionality of the proteins that is associated with the membrane. And here on the right hand side, you can see fluorescent microscopy photos of Bacillus subtilis, gram-positive bacterium. And this is just a map of different proteins that's in the cell or in the membrane. And you can see the treated ones in the red squares have a different morphology in terms of where you see the protein fluorescence. And specifically, if you look at the very bottom one, the nucleoid protein here is very much disrupted. The peptide also have an influence on DNA packing. So there's more than one target that we see when we treat microorganisms with these peptides. The current hypothesis is that the smallest antimicrobial moieties is amphipathic dimers. Now, why do we need amphipathic dimers? Of course, we need part of this molecule to interact with the hydrophobic core of the membrane, and then the other part of the molecule to interact with the more polar head groups of a membrane. So in this case, the highlighted part here is the hydrophobic part interacting with the membrane, whereas the part on the left-hand side is the polar part. These peptides have very complex activity, and it is most probably due to oligomerization. What you're seeing in front of you here is a mass spectrometry spectrum of a natural complex of these peptides that was produced. And you could see here on the left-hand side is the monomers, followed by dimers, trimers, and tetramers. We actually saw up to octamers using a mass spectrometry. And highlighting the dimers that are the putative active ones, we see quite a number of different dimers that is possible. And there is 225 possible dimers in the tyrosine complex if all the possible peptides are produced. This complexity actually helps with the broad spectrum of activity, as different types of dimers have different activities, as well as working against resistance because of the chemical and structural complexity. We mapped these dimers using ion mobility mass spectrometry. That is basically a wind tunnel in which both conformation as well as mass over charge ratio is used for separation of the different dimers. And in this case, it seems like size matters and not peptide identity. We have these discrete size groups where the interaction surface might be the important part in membrane interaction and for fitting in between phospholipids. Please note there's one that didn't fit into this, and this is the tryptophan-rich peptide that we spoke about previously with the unnatural production at high tryptophan concentrations. So what we also found is that the larger ones are more antibacterial, so with the average surface areas that are larger, so a larger interaction area, and the smaller ones are more antifungal. Working with these peptides are tricky and they are sticky, and they form all sorts of oligomers that we can trace again with ion mobility mass spectrometry. And this is just an example of a number of oligomers up to hexamers, and you can see also what is the average diameter of the interaction surface. So we are working between 2.2 nanometers to around about 5 nanometers in terms of how big these soluble oligomers are. And why is this important? This is important for us to know what is the size of the oligomers in solution, firstly for solution activity, but secondly as seeding oligomers for association with surfaces. These peptides are tricky to work with because they stick to each other and they stick to surfaces. Here is an example of a scanning electron micrograph showing the peptides on a mica surface. And you can see these little balls, and they're around about 23 nanometers average size, with a major group 22 nanometers in diameter. And if you imagine that one of these balls interact with a 1 micrometer organism, it would be the same as a .3 caliber bullet going through your body. There's about 1,000 peptide molecules in each of these little balls, and it might be that on a surface these balls are the active units. And this assembly was done in 15% ethanol. So if we use a fresh sample that is freshly made up and put that onto a surface, we get these little balls. But if you leave it too long in solution, the problem is you lose activity. And if you lose activity because of this oligomerization, it means that the peptide is less effective. So we can see this after 24 hours in solution. This graph shifted to the right hand side. That means that it is less effective. And if you look at on the left hand side, you can see there's already some larger balls, and the longer you leave it, the more of these larger balls will be visible, and these balls are less active in terms of its activity, and it also leaches peptide from the surface into these larger structures, which leaves the surface bare. At the moment, the surface is also still covered in this micrograph with a sheet of peptide from which these balls are growing. If we assemble these peptides from 60% ethanol, um, it is quite stable in solution, and it forms these films or sheets on the surface that is quite active. So the surface area is then more sheet-like and less nano ball structures, but it means that this is also quite active in killing microorganisms on a surface. These peptides and the solutions that they are in dictate the activity of materials like cellulose that's been treated with this. So if you look at this graph and you look at the green and the blue data points, you can see up to around 50 or 40% ethanol as a treatment solution worked very well, keeping the peptides quite active on the cellulose. But we are also looking at a mixture of peptides, which is the best option, or the phenylalanine rich complex in this case. And they work very well against Candida albicans. But the tryptophan rich and the mixed aromatic are not that effective, and they don't stick that well to the celluloses, and you can actually wash them off. So it has to do with both your peptide identity as well as the amount of ethanol that is used to treat the materials. And so that is something that needs to be considered when you are creating materials. We were able to create robust antimicrobial activity on cellulose. You could wash the cellulose quite a number of times, 12 times in this case that I am presenting here. The paper actually started to fall apart after 12 times of washing, so we didn't pursue it further. But for example, sodium chloride, acid washing, as well as 100% organic solvent washing, did not remove the peptide from the cellulose. We could only remove it with a basic solution, triethylamine, or a 50% organic solution, 50% acetonitrile. And in this case, we only removed about 40% of the activity from the paper. So this is quite robust. These peptides are also quite active after a time. For example, we could keep these celluloses for up to 2 years and only losing about 40% of the activity over 2 years, which is quite significant in terms of keeping a surface sterile for such a long time. We have treated many different materials for the packaging industry, and on the left-hand graph, this is for food packaging. That's why we tested Listeria monocytogenes, and these are all biodegradable packaging, polylactate and cellulose type packaging. And you can see here we killed more than log 6 Listeria monocytogenous cells per square centimeter. And then in the middle and on the right-hand side are different cellulose types of materials combined with cotton or polyester. And in this case, we also used 7% isopropanol and 70% isopropanol in order to treat these different materials. It seems like the 7% isopropanol was less effective depending on the type of material, whereas 70% isopropanol was more effective. But we still had more than log 6 killing of Staph aureus on all of the materials. And this differences also help us to decide on which application we're going to use, an organic solvent dip or spray application or a water dip application within the already processed materials that's going to be treated. So there is a potential in commercialization of treatment solutions in order to get these materials to have some sterilizing activity. So we think that we have dimers in solution, and then they absorb and into metastable structures on celluloses and polymer surfaces, and then they are released in the presence of pathogens, interact with the bacterium or the fungal cell, and in this way kill or lyse these cells. Thank you for your time. I just want to acknowledge all my funders, my postgraduate students and postdocs in the BioPep group, and my collaborators. Thank you very much.